Hello everyone, welcome to the short lecture concept. Today I am going to discuss about the second step of glucose oxidations, that is citric acid cycles. It is also known as the Krebs cycle or tricarboxylic acid cycles. It is fascinating me because it is a very interesting. If you learn it deeply, it is also fascinating. It will be also fascinating you. So the Krebs cycle, we have read it from our standard 10, okay, so as well as 12th, as well as in masters. So what is the new about this Krebs cycle? Okay, so Krebs cycle, it is the second step of glucose oxidation, which is takes place inside the matrix of the mitochondria, and it was first discovered by the Hans Krebs, hence its name is Krebs cycle. So in this cycle, there are 8 steps in Krebs cycle, right? From the 8 steps, there are many components which are directly or indirectly moves to other biochemical pathways, okay, to run the other biochemical pathway, which is very essential for the building of a cell. So the one question is came, if the components are moved to other biochemical pathway, then who will replenish these molecules, right? I'm saying that you are working in your home, right? So somehow someone called you that you have to join this work, right? So right, you are in the home, right? You are working some important things in home. If you go outside of the home, so you must have a substitute, right? So you should engage someone so that you will be leave from the home. So these things also happening in the tris carboxylic acid cycles, right? So there are many components act as a precursor for the synthesis of other biochemical or other molecules. So when these molecules are moved from the cycle, some other molecules which replenished this or to continue to restore these cycles. What are the different other molecules? How it will restore? So today I will be discussing about that. So let's move to the next, the replenished reaction pathway. So it is the complete picture of this replenished pathway or the reaction pathway, right? So you see that here the citrate, it is generally in some living bodies, living organisms, we isolate the citrate for commercial uses, right, for the production of a fatty acid, for sterols, right? And also alpha ketoglutarate, it is generally responsible for the synthesis of a acid that is called amino acids, that is the glutamate, right? So if alpha ketoglutarate, it is synthesized the building block of proteins, that is glutamate, then who will be replenished this alpha ketoglutarate? Then who will be synthesized this alpha ketoglutarate to replenish or restore these reactions? Will be discussed later. So now you should understand about which components are generally responsible for the other biochemical reactions. So first is the citrate, which is used for the synthesis of or the precursor of these sterols, and another one is the alpha ketoglutarate, which is directly or is a direct precursor for the one amino acid, acidic amino acid, that is the glutamate, and it is also act as the its carbon skeleton of alpha ketoglutarate. It is also responsible for the synthesis of purines and also other amino acids like proline, arginine, glutamine, right, which are the amino acids which are essential for our cell. The next is the succinyl CoA, and the succinyl CoA, it is also the precursor. It is the direct precursor of the formation of a porphyrin ring of a heme. Remember the heme that is present in our blood, the heme which is present in the hemoglobin, the heme which is present in the myoglobin, right? So For the synthesis of hemoglobin, for the synthesis of myoglobin, so heme is required for the carrying the oxygen. So this heme group, it is coming from the succinyl CoA, right? And another precursors which also synthesized from the citric acid cycles, that is the malate. Malate is the restore, which restore it, right? So next is the oxaloacetate. Oxaloacetate, it is the one of the precursor for the synthesis of aspartate amino acids, right? So if these molecules are participate in other biochemical pathways, then who will restore it, right? That's like the examples, you know. So it will be replenished by or restored by other biomolecules, which are the also the components of other biochemical pathways, right? So for example, if oxaloacetate is go to the synthesis for aspartate, then it will replenished by an enzyme is called a pyruvate carboxylase, right, which convert pyruvate into the oxaloacetate. It is restore the oxaloacetate. It is very simple manner. Okay. So then another question came, if pyruvate get converted into oxaloacetate, then who will continue the cycle? If all the pyruvate is converted into oxaloacetate right here, then who will continue the cycle? Because pyruvate is required for the fast product, required for the continuing the cycle, that is formation of acetyl CoA, right? You know, it is catalyzed by an enzyme is called pyruvate carboxylase, right? You know, pyruvate carboxylase, it is a positive modulator. It is an enzyme which is modulated by the oxaloacetate, right? You know, if oxaloacetate are very less, right, so then pyruvate converted into the pyruvate carboxylase, right? Because it is required to some pyruvate should be converted into oxaloacetate, right? If oxaloacetate amount is very high, then it positively modulates the pyruvate carboxylase activity. Please don't synthesize oxaloacetate. So it is inhibited so that the pyruvate is not converted into oxaloacetate, right? So then the cycle is continue and run. This is the beauty of oxaloacetate modulations. And next, it is not only restored replenished by pyruvate carboxylase, it is also restored by the gluconeogenesis steps. That is the, you know, oxaloacetate, it is converted into phosphoenolpyruvate and it is converted into glucose in gluconeogenesis steps. But the phosphoenolpyruvate, which is the ninth step of the glucose oxidation reactions, it is also written or converted into the oxaloacetate. This is by the phosphoenolpyruvate carboxylase. So oxaloacetate, it is the master switch or the regulator. When it will be synthesized or not, it depends upon its concentrations. It modulates the enzymatic activity. Okay, so one of the molecules get restored, that is the oxaloacetate, and is the regeneration molecules. You know, second molecule which also be restored, that is the malate. Okay, so malate is also restored by the pyruvate, right? You know, pyruvate is the end product of a glucose oxidation. So if malate concentration is very less in the cyclic cycles, so some of the pyruvate is converted into malate in the presence of a malic enzymes, right? So and the other molecules like citrate, alpha ketoglutarate, succinyl CoA, so it will, the work is going on. So how it will be restored? So right now these two molecules are restored by the different enzymes that we understood right now. So that's all about how the Krebs cycle work and how it replenished its components. Okay, so thank you everyone. So thanks.